Hello friends, welcome to my channel. Today we are going to discuss about the microsporogenesis. In this topic, we are going to discuss about what is plant embryology, structure of flower, a typical structure of anther, different layers of anther wall, functions of anther wall, the process of microsporogenesis, and development of male gamete. So let's see first what is the plant embryology. So it is a branch of biology which deals with study of embryology. But embryology not only includes the study of embryo but also it includes the process of microsporogenesis, megasporogenesis, development of male gamete, development of female gamete, and the fertilization process. So let's see first what is the flower. So flower, it is defined as a modified shoot as it is developed on the stem region. It is formed from different types of meristematic cells. So let's see what are the parts of flower. So flower, it shows the presence of the stalk. So the stalk of flower, it is called as an pedicel. The whole flower, it is based on a disc-like structure. That disc-like structure, it is called as an receptacle or the thalamus. The first oral of a flower is called as an calyx, which is usually green in color and helps in the process of photosynthesis. The next oral to the calyx is called as an corolla, which is generally colored in nature. And this corolla helps in the process of pollination. The next oral to the corolla is called as an androecium, which is a male reproductive organ of a flower. The androecium is followed by the gynoecium. It is a female reproductive organ of a flower. So according to presence of that four orals, the flowers are going to categorized under the four types, that is a complete flower. When the flower is complete one, whenever a flower shows presence of all the four orals, that is calyx, corolla, androecium and gynoecium, then that type of flower is called as an complete flower. Then next comes the incomplete flower. If a flower show absence of any one of the oral, then that type of flower, it is called as an incomplete flower. So that incomplete flower either show absence of calyx or corolla or androecium or gynoecium, any one of the oral. The next category, it is called as an bisexual flower. What is bisexual? If the flower shows presence of both the sex organs, that is androecium and gynoecium, then that type of flower, it is called as an bisexual. Then next comes the unisexual flower. If the flower shows presence of either gynoecium, that is the female reproductive organ, or androecium, either the male reproductive organ, then that type of flower, it is called as an unisexual flower. So before starting to the process of microsporogenesis, we must know a typical structure of anther. So see that this one is the typical structure of anther. This typical structure of anther shows the presence of the stalk, and the stalk, elongated stalk of that anther, it is called as an filament. The tip of filament, it shows presence of sac-like lobe structure. This one, it is called as an anthers. These anthers are developed by the apical and lateral meristematic cells. These apical meristematic cells and lateral meristematic cells undergoes division. After division, it can form a primordia-like structure, and that primordia, it will develop into the anthers. So see that these two anther sacs or these two anther lobes, they are connected by the tissue. It is called as an connective. When we take the tiers of anther, we are going to observe the structure like that. These are the four pollen sacs. So the formation of that four pollen sacs, that process, it is called as an microsporogenesis. So see that the typical structure of anther, it shows presence of three parts, that is the filament, which is the stalk of anther, anther lobe, these are the sac-like structure which are present at the tip of the filament, then the connective. These two anther lobes, they are connected by the tissue called as an connective. So this is about the typical structure of anther. Then next we see, when we take the tiers of anther lobe, we are going to observe the structure like that. The structure shows presence of four pollen sacs, and in that four pollen sacs, the numerous number of microspores are present. So see that the first layer of that microsporangium, it is called as an epidermis. This layer, outermost layer, it is called as an epidermis. The layer of epidermis, it is followed by the endothecium. This endothecium layer, it is followed by the middle layer, and that middle layer, it is followed by the tapetum. And these are the four pollen sacs. So this is the tiers of anther lobes, and this one, this part, this portion, it is called as an connective. So let's see one by one the anther wall. So as we studied earlier, so see that the first layer, that is epidermis, endothecium, this middle layer, and tapetum, all these four layers, they are together called as an anther wall. So one by one we are going to discuss about the layers of anther wall. So the first layer of anther wall, it is called as an epidermis. If you observe the cells of that epidermis, then cells are isodimetric. They show presence of prominent nucleus, dense cytoplasm. They are without intracellular spaces, and as it is an outermost layer, so it helps or it gives protection to the inner layers, as well as it prevents the cells from the environmental shocks. Then the next layer of tapetum, it is called as an endothecium. So if you observe the structure of cell, the cells are oval in nature. They show presence of cytoplasm, nucleus. Here the intracellular spaces are present, and the most important feature of that endothecium is they show presence of fibrous bands. So this layer of endothecium, it is hygroscopic in nature. It can accumulate the water, and whenever the conditions are favorable, by accumulating more amount of water, it gets burst and helps to liberate the pollen grains outside. So this is the function of that endothecium. Then next layer of the tapetum, the next layer of the anther wall, it is called as an tapetum. So this tapetum, it is the innermost layer of anther, as well as this tapetum is called as an physiologically important layer. So see that if you observe the structure of tapetum, the cells are either pentagonal or hexagonal in shape. They show presence of cytoplasm and nucleus. Then they stores the nutrients inside it. If you observe the tip of that cell, it shows presence of small opening, osteole-like opening. So through that opening, all the nutrients, they are provided towards the developing pollen sac or developing pollen grains. So this is the main function of tapetum, that is to provide nutrition. So see that these four layers together, it is called as an anther wall, that is the epidermis, endothecium, middle layer and tapetum. Then next we see the functions of each layer. Epidermis, as it is an outermost layer, it protects the cell or it protects the pollen grains from the environmental shocks, as well as it can avoid the loss of water. The next layer, that is the endothecium, as it shows presence of fibrous bands, so it helps in the discharge of the pollen grains by bursting the anther wall. Then the next layer, it is called as an tapetum, as it shows the presence of nutrients, so it helps to provide nutrients towards the developing pollen grains. So let's see what is microsporogenesis. So see that actually the microspores, they are developed from a type of cells, they are called as an sporogenous cell. So before the development of that pollen grains, it is a mass of sporogenous cell. Now, not all the sporogenous cell have the ability to develop into the pollen grains. So generally one or two of the sporogenous cell, they are going to enlarge itself by accumulating more cytoplasm and nutrients from the surrounding cells. So see here, this is an enlarged cell. It is called as an active cell, and further it acts as an archisporial cell. It get enlarged itself by accumulating more amount of cytoplasm from surrounding cell, as well as it can accumulate nutrients from the surrounding cell. Now this enlarged cell, it becomes active, it functions as an archisporial cell and undergoes the transverse division. After the transverse division, it can form two layers, that is the upper layer or upper cell, it is called as an primary parietal layer, and the basal cell, it is called as an primary sporogenous layer. So see that this primary parietal layer again undergoes lots of anticlinal and periclinal division to develop the wall of anther, that is to develop the epidermis, endothecium, middle layer and tapetum. While in some of the plant species, the primary sporogenous layer, it directly functions as a microspore, or it can undergo further divisions to develop numerous number of microspores. So this development of the microspores, that process is called as an microsporogenesis. So let's see that. It is a mass of sporogenous tissue. Only one or two cells have an ability to develop into the microspores, and remaining other cells, they act as a nutrition for the developing cell. Then this single sporogenous cell, it get enlarges by accumulating more cytoplasm and nutrients from the surrounding cell, and that enlarged cell, it functions as a microspore. Mother cell are called as an archisporial cell. This archisporial cell undergoes transverse division to form the primary parietal layer and primary sporogenous layer. So this primary parietal layer again undergoes lots of anticlinal and periclinal division to develop the wall of anther, and the sporogenous cell, according to the type of species, either it behaves as an microspore or it can undergoes division and then develop lots of microspores. So see that after the process of microsporogenesis, there are different types of microspores which are formed according to type of species. So see that what are the types of microspores. So the first type, it is called as an tetrahedral. Second one, it is called as an isobilateral. Third one, according to their structure, it is called as an decussate. This one, it is called as an T-shape, and this one is the linear, in which the cells lie one above other, called as an linear. So these are the different types of microspores which are developed after the process of microsporogenesis. So whenever the conditions are favorable, then the endothecium layer, tapetum layer, they are going to accumulate more amount of water, and with the help of that fibrous bands, the wall of anther, it get burst, and after bursting of the anther wall, the pollen grains, or you can say the microspores, they are liberated outside. So this is the structure of the microspore after liberation or after liberated outside from the anther wall. So this microspore, it shows the presence of two layers, that is the outer layer, it is called as an exine. So depending upon the species, it may be smooth or spiny in nature. Then the second layer of that microspore, it is called as an intine. It is thin in nature. Then that pollen grain shows the presence of small gaps. So that gaps, they are called as an germ pores, and from that germ pores there is formation of pollen tube. So next we see the development process of the male gamete. So see that this liberated pollen grain, which shows the presence of exine, entine, it undergoes transverse division to form two cells. The upper cell, it is called as an vegetative cell, and the lower cell, it is called as an generative cell. This vegetative cell undergoes division to form the pollen tube. It can form the pollen tube nucleus, while the generative cell undergoes division to form the two male gametes. Then during the lateral developmental stages, this pollen tube nucleus, it get degenerate, and the pollen grain shows only presence of the pollen tube and that two male gametes. So this formation of two male gametes, that process, it is called as an development of male gamete. So see that after the liberation of microspores, it divides transversely to form two cells, that is generative cell and vegetative cell. Vegetative cell undergoes division to form the pollen tube and pollen tube nucleus, while the generative cell undergoes division to form the two male gametes. So during the lateral developmental stages, the pollen tube nucleus it get degenerate and the male gamete shows presence of pollen tube and two male gametes. So this is the process of development of male gamete. So see that after microsporogenesis, the microspores are developed, and when the conditions are favorable one, the anther wall get breaks, and after breaking, the pollen grains they are liberated outside, and the layers like endothecium and tapetum they helps in the liberation of microspores. The developed microspore it shows presence of exine and entine. Exine it is thick in nature, while the entine it is thin in nature. While this exine shows presence of small pores to come out the pollen tube, and that small pores they are called as an germ pores. So here the liberated microspore divide transversely to form two cells, that is vegetative cell and generative cell. The vegetative cell undergoes division to form the pollen tube nucleus, and generative cell it divides to develop the two male gametes. Further, the pollen tube nucleus it gets degenerate, and the mature pollen grain it shows the presence of only the pollen tube and two male gametes, and that pollen grain it is ready for the process of fertilization. So this is about the microsporogenesis. If you like this video, please like, share and subscribe my channel. Thank you.